Okay, it's my pleasure to introduce Dr. Tom Prisinzano, give his talk. Dr. Prisinzano received his BS in chemistry from the University of Delaware and a doctorate in pharmaceutical science from Virginia Commonwealth University in Richmond. From 2000 to 2003, he was an Intramural Training Award Fellow at the National Institute of NIDDK. That's where I got some of my funding from. I got to know Tom at the University of Kansas. He was in medicinal chemistry, along with Dr. Emily Scott, who will be speaking later this year, and myself. I was in pharmacology and toxicology. But these folks formed the core of my peer group while I was at University of Kansas. And I got to tell you, they set a high standard of behavior in terms of laboratory science and collegiality. I found them to be very inspirational in many, many ways. Dr. Tom is incredibly well-funded and doing some very interesting research that, by the way, interfaces with some of the things we're doing in Joplin. So it's a pleasure to welcome Dr. Tom Prisinzano to give his talk today entitled Chemical Approaches to Addressing the Opioid Epidemic. Tom, take it away.
All right, great. Thanks, Jeff, for the kind introduction, and it's great to see old friends and tell you about some of the fun stuff that we've been doing as it relates to here in terms of the the opiate epidemic. And so it really goes without saying that, I'm sure that, even though now we're thinking more of COVID as being an epidemic or a pandemic in this case, but realize that there's been a rise, at least in terms of the opioid epidemic, which has unfortunately hit a lot of towns here in Appalachia and certainly throughout the country in terms of the rise. And so obviously the problem is that if we look here at the screen, people have moved from prescription opiates to now injecting and ensuring the rise of heroin, and as I'll get to a little bit later in the talk, into fentanyl and related compounds. So overall, in terms of what my research interests are, is looking at specifically pain and drug abuse. Specifically, how do we get addicted in those particular cases? And so if we can understand how we can become addicted in general, then we can then look and use science, hopefully using the process of medicinal chemistry to try to come up with treatments to treat drug abuse as well as to stop drug addiction in general, whether that be substance use in any many substances, as well as the better treatment of pain. And so it's a little bit of background when we talk about in terms of opioids and pain. Opioids have been the mainstay for the treatment of pain for centuries. And we can look at pain treatments like opiates can be divided into a couple different categories. The first being partial agonists, things like, to some extent, at least depending upon how you test, morphine as a partial agonist versus a full agonist, which can be highly potent compounds, so things like fentanyl, and sort of mixed agonists and antagonists. And these are going to be compounds which have a combination of both agonism as well as antagonisms. And if we look from a pharmacy sense and we look at other agonists and try to divide them down into further strong and weak, when we think about strong agonists, there are going to be things like morphine, oxymorphone, methadone, levorphanol, certainly fentanyl-related compounds, and oxycodone. Weaker agonists are going to be classified as things like hydrocodone, codeine, and tramadol. A lot of this has to do with in terms of the metabolism of these particular agents. Now, really what separates opioids from other types of say, non-steroidal anti-inflammatory agents or other treatments for pain is that there's generally no ceiling for their analgesic effectiveness. And so, being around first at the University of Kansas and now here in Kentucky, being around farm, and oftentimes you have farm accidents where you have sort of catastrophic injuries. And if you think about somebody who has their arm cut off or someone with these types of really major injuries, you can realize that 800 milligrams of ibuprofen isn't going to be enough to cover the pain. And so therefore you need something a lot stronger, which is where you're going to need your opiates in this case. Now the problem is that the opiates themselves are great in terms of treating away that type of pain, but they're associated with various side effects. Namely, this is going to be tolerance, where one is going to have to escalate the dose to produce the same level of effect. Respiratory depression, which is how almost all opiate addicts die, and because in sort of the good news, bad news in case, when one takes an opiate, you're certainly going to relieve pain, but if you get the dose too high, it also suppresses respiration, and so if you suppress respiration, this eventually leads to death. Now, another one from more of a pharmacology-based one, or at least in the clinics, is actually that of constipation. And so what ends up happening is that an opiate person eventually has the problem where they actually turn off the bowels. Now, this can be used therapeutically in the case of diarrhea, and most of your antidiarrheal agents do function as peripherally restricted opioid agonists because they turn off the bowels. But as I mentioned, we're sort of in the wave here in terms of the opioid epidemic, and this really began back in approximately 2000. And so what this has is in terms of the opioid prescription in this case, what we can see is that we have here in terms of purple, we see the rise of prescription opioids, which are used for the treatment of pain, leading towards misuse. And so roughly about in wave 2, where we see the rise of heroin coming back in sort of being flatlined in this case around 2010 up to its peak levels now, which we know is sort of resurgence. And really where you see things take off is actually now is really the rise in synthetic opioids and things like fentanyl-related compounds, which are finding their ways into the street. And we'll talk more about this a little bit later. But just a few numbers to give you. The first is that the National Institute of Drug Abuse estimates that approximately 130 people die in the U.S. every day as a result of using opioids, making them the #1 prescription medication that produces death in this case. And it's been, obviously, there's been various ways that you can estimate in terms of the economic burden associated with the opioid epidemic. At least one measure is that you can estimate that it's approximately $80 billion a year as a result of prescription opioid misuse. But as we know, the pain is probably the #1 reason that people come into the emergency rooms for the treatment of that. And so common types of pain are obviously going to be neck pain, the lower back pain, or even that the pain associated with severe migraines. And so realize in terms of one of the tenets, at least in terms of the emergency room, is obviously the treatment of pain. And so really what we need better medications to treat pain. So going back historically, really one could say that morphine is really the first pharmaceutical. And the fact that this beautiful structure shown here as a T-shaped morphine molecule going back to the early 1800s, when it was first isolated by Sertiner in 1803 and produced in this game. And morphine gets its name from the, Morpheus, the Greek god of sleep, because as we know as a traditional narcotic or opiate, it's one that actually produces sleep and produces sedation known there. So here's just showing you some of the things. Now, believe it or not, we do actually still get morphine-related compounds actually from extraction of the natural products. And so this is actually showing you how the opium production, or how we're actually get raw opium. And so this is actually shown in the fields of Afghanistan. And so what these particular farmers are doing is the process that's called milking the poppy. And what they're doing is they're taking this shovel or this particular spoon-like instrument, and they're actually going and touching each one of the individual poppies in this case, and they're creating a lance. They're lancing it and opening up the unripe poppy, and this is going to produce a latex, and they'll come back about a week later and they'll scrape it off, showing you the case here when they scrape it off, and showing you what looks like raw opium. And so with raw opium from this case, we can predominantly get 3 types of opioid analgesics or opioid compounds. We get a mixture of morphine, codeine, and the more important one, which is actually that of thebaine. So thebaine itself is not really analgesic in and of itself, but it's the main precursor for almost all of your 14-hydroxy compounds. So things like oxycodone, buprenorphine, and Narcan or naloxone actually come from thebaine rather than morphine or codeine specifically. And so while I'm certainly not going to say that the Taliban was great, one of the beneficial things at least happened when the Taliban was ruling Afghanistan is they really clamped down on the opium trade in this case. And so, as you know, we've seen a rise here as this really leads to the problems associated with the opioid epidemic as people transition from prescription opiates eventually back into heroin. Now, opioids and related compounds actually exert their biological effects upon opioid receptors. And so the endogenous ligands for opioid receptors are neuropeptides. But at least in some interesting studies, morphine has actually been shown to be produced in various types of cells biologically. And the main opioid peptides are enkephalin, endorphin, and the kappa-comparing type in terms of dynorphin A. There's 3 major types of opioid receptors: mu, delta, and kappa. And there's evidence for some pharmacological evidence for subtypes of each of these receptors. We haven't actually found the gene products for those yet. Most of your clinically used medical narcotics all work as agonists at the mu opioid receptor and do have varying amounts. So morphine and the mu receptor actually gets its name from morphine. Now, when we think of opioids, we definitely think about their effects, at least in terms of analgesia, but they're also involved in a number of pharmacological other processes, one of which is the immunological response. And so this is part of the reason for the major public health threat associated with opiates, because what happens when one chronically takes opiates, you suppress the immunological system. And by doing this through IV unsafe practices, you exacerbate the spread of various diseases, in this case, HIV, as well as drug-resistant tuberculosis. Now, opioid receptors themselves are members of the G-protein coupled receptor superfamily or 7 transmembrane receptors, which we'll talk a little bit about later. And this is actually a picture of the opium poppy done by Linnaeus. As I mentioned, the endogenous peptides for opioid receptors are these two particular ones. Two ones I'll draw your attention to are met-enkephalin and leu-enkephalin. These were the first two that were actually discovered by Hughes and Kostelich back in 1975. And they're very simple structures, and they only vary in terms of the terminal amine, terminal structure shown here at the end, whether it's a methionine residue or a leucine residue in this case. And so without going into too much medicinal chemistry, as it turns out, its critical residue here is this phenolic hydroxy group, which is thought to interact in the same spot as actually the phenolic hydroxy group associated with morphine. But as you know, we've been studying opioid receptors and opioid compounds for the better part of 100 years, and some of the selected ones I'll draw your attention to, which we think about in terms of the treatment of pain. And the first is actually that of hydromorphone or dilaudid, which is a derivative of morphine, where we actually reduce the 7-8 double bond and oxidize the ketone up to a corresponding ketone. And so with hydromorphone, it's approximately 7 times as potent as morphine in animal tests. When we think about OxyContin or oxycodone in this case, or otherwise as we refer to it here in Kentucky as hillbilly heroin, oxycodone in this case is the preferred one, where it contains a 14-hydroxy group. So structurally, it's related to naloxone and naltrexone, which are opioid antagonists, and the main difference in terms of opioid structure-activity relationships is the nitrogen substituent. Now, it is possible to generate compounds which are considerably more potent than morphine-related compounds, and probably the best example is actually that of atorphine, which is incredibly potent as an opioid analgesic. It's approximately somewhere between 10,000 to 20,000 times as potent as morphine is as an analgesic. And this particular compound is only used in large game animals as a compound to knock them down as a sedative in this case, because the lethal dose of atorphine is about 2 milligrams and the effective dose in humans is about 1 milligram. So therefore, from a therapeutic index standpoint, it's actually quite small. And so when atorphine is actually shipped out, it comes out with also a corresponding antagonist as a package to allow for pre-injection in this case. So it's just an example showing you some of the structure-activity relationships amongst these compounds. So go a little bit in terms of the history associated with opioids in this. So as I first mentioned, morphine was first isolated by Serturner in 1803, and it was the late British chemist Sir Robert Robinson first proposed the structure in 1925 of morphine. And so realized that morphine entered into the pharmacopoeia of multiple countries somewhere times in the 1800s, and so they did this without actually knowing its chemical structure of the main active component. It wasn't until 1954 when we actually thought at least the structure of receptors of morphine and related compounds interacting with a receptor was done by Beckett and Casey in 1954. It wasn't until 1955 that the actual structure of morphine was confirmed through the total synthesis by the efforts of Marshall Gates and Giglio Schutte. Fast forward 20 years or so, it wasn't until 1973 when Candace Pert and Saul Snyder actually showed the actual presence of opioid receptors in nervous tissue, and actually in the rat brain in 1973 in a seminal paper in the Proceedings of Casual Anatomy of Science. As I mentioned, the endogenous ligands for opiates weren't discovered until Hughes and Kostelich and related and coworkers in Nature in 1975. Here at the University of Kentucky, William Martin first proposed that multiple opioid receptors were actually a thing rather than just a single opiate receptor back in 1976. In 1993 was when we first cloned the mu opioid receptor, and finally in 2012 is when we finally got the actual crystal structure of the mu opioid receptor in this case. So realized that it was a little almost 200 years from the first isolation of morphine till under knowing at least in its biological target in terms of actual crystal structure, which was actually done by Brian Kabilka and coworkers. So all that being said so without that background, how does one begin to potentially combat the opioid epidemic in this case? And I would argue amongst the potential chemical options that you can do is the first thing you can do is actually you could limit the supply of opiates. And the Drug Enforcement Administration has actually done that. So what they've done is to begin to limit the supplies and limit the ability to buy or even have the supply of opiates from where we actually get it overseas. And the DEA has actually dropped the number of 20% on top of it, 25%, which was seen in 2016. So that certainly is a strategy, but it doesn't really develop, at least in some of the underlying problems, where people who are already addicted in this case. So realize there's another way to do that is potentially to develop agents that can be used as for the treatment of pain without the substance use disorder potential. Because we do know that opioids are essential as pain medications for the practice of monomention, but they do have potential for misuse. So either you could sort of broadly characterize this as sort of find analgesics that don't have an abuse liability, which would be great. So it's what we refer to this as the holy grail of analgesics, where we could find ones that are just as efficacious as morphine, but don't have the side effects associated with traditional opiates. So don't start. And the other one would be that of actually to be able to find medications to help people come off. And so there's a number of agents to treat substance use disorder or opiate use disorder. So things like methadone, buprenorphine, and naloxone are certainly useful, but they're not optimal for all patients. And so we do need some better medications to do that. And so efforts in my lab really sort of taking two different tacks. So the first one I want to talk about is to potentially find agents to treat substance use disorder. And so the approach we've taken in my lab is nearly all non-peptide opioids are derivatives of morphine. And I'll show you this in a second. And so there's a good percentage that they're likely to share common side effects. These are obvious, as I mentioned before: tolerance, respiratory depression, constipation, addiction, or substance use disorder. And so what we wanted to do, at least, is to try to find novel agents that might serve as templates for the development of opioid receptor compounds, but don't have a morphine-like structure. And hopefully, if we could do that, we could study them and investigate their structure-activity relationships. And so as I mentioned, here are sort of the structural classes of opioid analgesics. So we have the 4,5-epoxymorphinanes like morphine. If you excise or remove the epoxy bridge, you can generate a compounds of the morphinane scaffold. So an example of this would be a compound like levorphanol. And then if you actually remove and further excise this other 6-membered ring, you can generate a series of compounds called the benzomorphans. And so these are the example ones of like cyclazicine or pentazocine are two of the ones of this type. If one opens up again this particular 6-membered ring system, you can create a series of compounds called the 4-analytyl piperidines. And if one does that, you can generate a series of compounds like fentanyl and related compounds. And furthermore, if you actually open up this 6-membered ring, you can get ring open structures of things, the 3,3-diphenylpropylamines, of which methadone is probably an example for that. And if one does a different disconnection related to that of morphinanes and remove a different 6-membered ring and transpose the nitrogen, you can get a series of compounds called the 5-phenylmorphans. And so we have opioid analgesics of these general types. Now, tapentadol has been marketed as a new opioid analgesic related to this particular structure, but I've drawn it in a particular fashion. As you can see, through a different series of disconnections, it looks an awful lot like what's been seen before previously in terms of other opioids. But really, I want to draw your attention to structure in gold here, which is actually very different from that elicited from the other compounds. And the first, that in fact, it contains no basic nitrogen, which would be a foremost absolute critical requirement for interaction of opioid receptors. And this class of compounds are called neoclaridanes, and they're more closely structure related to cholesterol and related compounds, natural products. And so this particular compound is a compound called salvinorin. And a work done by Brian Roth showed that this particular compound actually bound selectively to the kappa opioid receptor, one of the 3 types of opioid receptors, mu, delta, and kappa. And it turned to be a potent kappa opioid receptor agonist in terms of binding, and the binding could be shifted rightward with opioid antagonist naloxone. And it produced effects similar to that elicited by standard other kind of kappa opioid receptor ligands, like a compound was developed at Upjohn called U69593. And so this is actually as the main premise at the beginning of our work, at least in this particular compound. But really what intrigued us as well is that Salvia divinorum is a mint plant native to Oaxaca, Mexico, which is the area down here in the bottom, close to Guatemala, in the area where Mexico becomes quite, the distance between the Atlantic and the Pacific becomes quite small. And so the main active component of the plant is Salvia divinorum is salvinorin A. And it's used by the Mazatec Indians as a hallucinogen. And so what it does is it's a sacred material that's tightly regulated down there. And so what it does is it produces a visionary trance that allows the shaman to see what steps need to be taken to cure the individual patient. And so what ends up happening is you can have a semi-magical disease, which roughly translates called Prisinzano de Borrego, which roughly translates to swollen belly. And so the way that one would overcome this type 1 is you would actually have to smoke Salvia divinorum, and to do that to alleviate the pain associated with a swollen belly. And so the predominant use, at least in terms of salvia divinorum, is it's predominantly smoked, or it can also be used as a quid for bicuccal absorption, much like you would use chewing tobacco. It produces vivid hallucinations and out-of-body experiences. And it produces a hallucinogenic experience which is very different from that sort of classical hallucinogens, much like, say, 5-methoxytryptamine or LSD. And it also has a very low abuse potential, which means it's similar to that of the psychoactive substances like your classical hallucinogens. Now, just to give you an idea in terms of the potency of salvinorin A, a smoked dose of somewhere between 0.4 to 21 micrograms per kilogram is enough to produce hallucinations for an hour. So this particular compound is among the most potent naturally occurring hallucinogens that we know of. So just to show you, at least from a chemistry perspective, is if we look at sort of hallucinogenic substances, things like delta-9 THC, if we look at atropine, which is going to function as a component of datura stramona and other belladonna alkaloids, phencyclidine or PCP, ibogaine, which is present in the West Africa show of Jabura iboga, lysergic acid diethylamide or LSD, or mescaline or dimethyltryptamine, you can clearly see that salvia divinorum's main active component, salvinorin A, looks nothing like any of the other hallucinogenic substances. So from a chemistry perspective, this is pretty fun and pretty cool. So now we have a unique structural scaffold that appears to interact with opioid receptors, and in the same vein appears to also produce hallucinogenic effects, very different than other known hallucinogenic substances. So as a medicinal chemist, one of the things that we're interested in is sort of this design cycle, which we'll talk about, is sort of the cycle of where we design novel agents, we make them, we eventually test them, and then analyze them, and then sort of the cyclic cycle to find something that's good, and eventually in the process of risk assessment, get compounds which can be put into clinical development. And so my lab is very much interested in sort of the process of lead optimization, where you sort of have this iterable process of improving compounds, and then secondarily to try to advance them into various models to de-risk them before going into clinical development in this case. And so I'll talk a lot about this sort of DMTA cycle as we relate towards salvinorin A as a lead molecule. So one of the first things we did is actually get salvia divinorum and related compounds. What we actually do is we actually extract salvinorin A from dried leaves of salvia divinorum. And this actually is quite funny because we can actually get these compounds actually delivered. They were commercially available on the internet, and they used to actually have a huge shop in downtown Lawrence, Kansas, where we could actually buy kilograms and just go downtown and have them shipped over to the School of Pharmacy. Once we do that, we can grind up the leaves into a fine powder and then extract them to get the main component, salvinorin A. Once we do that, we can then do a series of simple chemistry and change and make a series of derivatives, one of which is certainly we can take the ester here off and create another natural product called salvinorin B, to then produce a corresponding phenyl substituent here. And so the simple change of going from an acetate ester into a phenyl benzoyl ester, we get a compound which we'll call hercinorin, and we'll talk more about the biology of this in a second. And in addition, through a series of steps, we can convert this particular alcohol into a better leaving group of bromine, which then allows us to substitute with an azide group, which then allows us to then reduce the azide down to an amine. And then we can do the same types of substitutions to generate an amide derivative. The reason for that is we certainly know that amides are more stable than corresponding esters. So it gives us a better handle, at least to understand the compound, which is going to be more stable for biological experiments. So with this in hand, we made this series of compounds. Now, the way we evaluate our compounds for their effects at opioid receptors is to use this assay called the cyclic AMP hit hunter assay. So what happens is that opioid receptors are negatively coupled to the second messenger coupled adenylate cyclase. Okay, so adenylate cyclase being negatively coupled means it turns off the actions of the enzyme adenylate cyclase, which is involved with the conversion of ATP into cyclic AMP. Now, the way to stimulate the activity of adenylate cyclase is use a different natural product, forskolin. So if you give cells which have been shown to express opioid receptors with adenylate cyclase, you give forskolin, it stimulates adenylate cyclase, which then increases amounts of cyclic AMP, which, if you have a cyclic AMP detection method, can produce light. Okay. Now, that shows you here in green. So no opioid activation, I give forskolin, I get no inhibition of adenylate cyclase, I produce a high amount of light. Now, in the case of an opioid agonist in red, which binds to the kappa opioid receptor, what it does is it involves with it break apart the G protein, and the G protein alpha subunit goes and interacts with the adenylate cyclase. And if I also then treat these same cells with forskolin, I now have my opioid agonist, which is going to inhibit the actions of cyclic AMP. Cyclic AMP production, I'm going to produce less cyclic AMP and less light. And by difference, I can then get a measure as to how well this particular compound interacts with the kappa opioid receptor. All right, so much like golf, this is the lower the number, the more active the compound is in producing effects of the kappa receptor. This is nice because this particular assay is a functional assay. It's not a binding, per se, a binding assay. No radioactivity is necessary, and it's a pure functional assay, and it's going to use light for this case. And the nice part about this is that we can have cells which express each individual opioid receptor, mu receptor cells, kappa receptor cells, delta receptor cells. And so therefore we can get selectivity relatively easy by giving the same compound to 3 different cells pretty fast. And this is a commercially available technology which is developed by Eurofins out of St. Louis. Now, just to give you some sense here in terms of some selected structure-activity relationships, so dinorphin A is the endogenous kappa opioid receptor peptide. And so if we do that, notice it's quite potent at the kappa opioid receptor at 0.4 nanomolar. U69593 is a standard kappa opioid receptor compound, which is developed by Upjohn, which is a derivative of morphine. And look at salvinorin A, you can see this compound is exquisitely potent at the kappa receptor, even more so than the endogenous ligand. And we know that we can simply make simple changes, making a bromine or a methyl group or an ethyl group here on the aromatic ring. Notice that we can modulate the level of activity, which we can see. And furthermore, we can end up making a series of substitutions all the way down. If we make a judicious choice here of an ethynyl group, we can actually end up with a compound which is more potent than the natural product. Okay. And furthermore, we can see that it's exquisitely selective for the kappa receptor over the mu receptor in this case. Now notice that simple change I talked about before between hercanorin and hercamide. By putting that benzene ring, I can now switch the selectivity of the compound from being selective for the kappa opioid receptor like the endogenous natural product salvinorin A. I can make it actually now selective for the mu opioid receptor in this case, which is actually pretty interesting. And we'll come back to this in a few minutes. But one of the ways that you can actually target and develop drug abuse medications is you can do that by actually targeting kappa receptors. And so it's been shown that kappa receptors are involved in some of the abuse related of CNS stimulants, such as cocaine or methamphetamine. And so there's a number of ways that you can test drug abuse medications. I would argue there's sort of 2 good animal models that can do that. And one is sort of the gold standard, which is self-administration, which is exactly what you think. You give an animal a free choice between choosing, say, cocaine or heroin from saline, and as you might not be surprised to know that the animals like taking those particular substances, and so they'll self-administer one versus the other. Okay, and the second is more a model, at least in terms of what happens with an addict, which is actually that of the model of relapse. And so the problem is as you know, when people go to rehab, they eventually come out of rehab, they get cured, which I'll put in quotes, and eventually what happens is they live their life events change, things happen, they fall off the wagon, and they started using again. All right, we refer to that as relapse. Or in this particular case, we talk about it in terms of reinstatement of extinguished drug-taking behavior. So we can actually model this relapse pretty well. So what ends up happening is we take animals, in this particular here showing you a rat, which has a free access to a choice between cocaine versus saline. And so what ends up happening is the rat, in this case, or a mouse, actually looks and it actually chooses to take cocaine over saline. And the way we send the rat to rehab is simply to turn off the cocaine lever. No matter how many times they hit it, they're going to no longer be able to get cocaine in this case. So it's kind of brutal in some particular fashions. But what it does is they eventually learn that they can't tap the lever anymore, and they actually are no longer sort of dependent upon cocaine. What ends up happening later is then we can then simulate falling off the wagon by giving a larger dose of cocaine. And notice what ends up happening now is you see the reinstatement of this extinguished drug-taking behavior coming back even more so in this case in terms of statistically significant. And so if you had a compound which is going to block self-administration in the first place, it'd be for the first half. And if you're going to block reinstatement, it's going to be in the second half. And what this is going to show you in terms of this case is that we have analogs of the kappa opioid receptor of salvinorin A is actually able to block cocaine-induced reinstatement in rats. So we can actually block this reinstatement effect with a series of compounds. So notice we had the vehicle, and looking at it in a dose-dependent manner, we can actually block the cocaine reinstatement effects in this particular case, which is actually pretty interesting because we can potentially treat cocaine or methamphetamine abuse with these particular types of compounds, which is actually pretty interesting. Now, as I mentioned, part of the problems associated with kappa receptors are their side effects, and so one of which is actually that of sedation. So a joke amongst the field is a sleeping monkey or a sleeping rat self-administers no cocaine, all right? And so what you would want to do is you want to see that whether or not they have a sedative effect. So there's a multitude of ways that you could test this, one of which is actually cocaine-induced hyperlocomotion. And so if you give a rat cocaine, they're going to run around the cage to a higher degree in terms of this hyperlocomotor activity. If you give a sedative compounds, they'll stop running around the cage to the same extent. And what this is showing you is the series of compounds that I showed you on the previous slide have no effect compared to saline, in this case, not producing a sedative-like effect. Now, one of the other problems associated with kappa opioid receptors is they do have potential to produce depression. And so one of the ways that comes about is this case. And so we can do this in sort of an antidepressant model. And so one of the ways to do that is sort of the learned helplessness or forced swim test, which you take a rat or a mouse and put it in a glass of water. And what ends up happening is they don't like water, so they try to fight to get out of the glass. And eventually what happens is they learn to be helpless because they can't do anything. And so a compound which is going to be a depressant is one which is going to produce a decreased amount of climbing and a decreased amount of swimming and an increased amount of time immobile. And so your antidepressants would be the exact case. And so what you're showing here is a 16-bromo compound, has no statistical difference between vehicle in terms of climbing or swimming, and it's going to have no difference in terms of immobile time, suggesting that this compound has less depressive-like effects compared to other standard kappas. And I should mention that if you give the compound, say, like U69593, you will see increased immobility in this case. Now, in non-human primates, you can test in this particular compound in terms of sedatives. So obviously, the types of experiments that you can do in non-human primates are different than that elicited from certainly from rats or with mice. And so this is showing you in terms of a biomarker which is prolactin. So with a kappa opioid receptor agonist interacts with kappa receptors, you can see an increased amount of prolactin, much like with salvinorin A. And notice you can see a rightward shift in the dose response curve when you give a derivative of salvinorin A. Now, a compound which exists as a kappa partial agonist rather than a full agonist, you'll see that plateauing of this effect, much like Morphine, which appears to be a kappa opioid receptor partial agonist. Now, the interesting part is actually if you look at sedation, at least by using visual software and cameras in the non-human primates, you can see that the compound produces less sedation with a simple change in this case. So now we have two species which suggest that the compounds are producing less sedation, meaning that they're different kappas as a potential lead molecules towards the further development of agents to produce less addictive-like medications, at least in this case, or an anti-drug abuse medication. Now, the other thing I want to talk about is sort of these two compounds, hercinorin and hercamide, because what ends up happening is when we did salvinorin A-related compounds, these compounds switch their selectivity from being kappa opioid receptors to mu opioid receptor compounds, meaning that they had the potential to be better analgesics compared to that of the kappas. And so if we look at the X-ray crystal structure, what we're showing you here is sort of this green aromatic ring in the X-ray crystal structure. And hopefully what you can see is sort of the difference between sort of a perpendicular to the plane of the ring here in the top and a parallel fashion in the bottom in this case. And what we know is that we can further increase the selectivity for the mu opioid receptor if we have a conformation like the bottom compared to the conformation on the top. And so be able to do that is how do we design molecules to do that? And if we did that, we should have analgesics or mu opioid receptor compounds, which potentially could be better analgesics. And so one way it does that, at least in medicinal chemistry, is to actually now introduce a double bond. So if we do that, we create this particular double bond, we can generate a compound called hercinorin, whose structure is shown here. And so now, at least in terms of medicinal chemistry, we know that if we have the symbol structure between hercinorin and hercamide, those compounds have one stereochemistry. And if we then use different chemistry, we can invert the stereochemistry to create a different compound in this case. And so if you look up sort of my arm, we now have a possibility to have molecules as it relates to the plane of the ring. We can have molecules which are in the natural case, which are in the bottom. I can have molecules in the center, which are going to be perpendicular, or I can have molecules in the top. And so I have the beautiful sort of butterfly type of an effect. Now, what does this do biologically? So if we compare DAMGO, which is a selective opioid peptide for mu opioid receptors, and we look at morphine, we can see that they have high activity at the mu opioid receptor compared to that of kappa receptors. Now, as I told you before, hercinorin, which had that aromatic ring, I could switch the selectivity from the kappa receptor to the mu receptor, which is done here in terms of hercinorin, and the compound is about 10-fold less case. Now, if I change the stereochemistry of hercinorin to go to the opposite stereochemistry, meaning no longer has the natural orientation in the alpha position, but above the plane of the ring of the Declan system, I can see that compound is completely inactive at both kappa and as well as mu receptors. Much to our delight, if we actually get the sweet spot and find the compound which is now in the middle in the exact orientation, notice now we can get a compound which is 1.2 nanomolar and is actually incredibly potent and selective for the mu receptor over kappa. All right, well, this is all fine and well. Is it actually work as an analgesic? And so one of the problems originally with hercanorin was the fact that it didn't cross the blood-brain barrier, okay? And too much to our delight, if you look at compound 5, which is the compound hercanorin, notice that this compound does cross the blood-brain barrier. So this is actually in the hot plate test, which is exactly like it describes. And so what do you think? Much like you would burn your hand on a hot stove is how this actually works in terms of animals. What happens with the animals is that you put them on a hot plate, okay? And then as they might imagine, they get a noxious stimulus, and eventually at some particular times, they're going to jump off the hot plate, okay, from the latency time. If you give them an analgesic substance, they're going to stay on the hot plate longer, okay, eventually to some cutoff time, and eventually for tissue damage. Now, if we give an opioid antagonist like naloxone, okay, or Narcan, that'll block the opioid actions in this case. So if we look at, in this case, if I give morphine, okay, to the hot plate, I produce an increased effect, which then falls off over roughly a 2-hour time period. If I give morphine and naloxone, I completely block that antinociception action. Now notice hercanorin does not produce endospermin and it's stuck in the periphery. Now, electrolyte in blue with hercanorin, notice it produces an antinociceptive effect, which falls off the 2-hour time period, and we can see this in a dose-dependent manner. And if I pre-treat with naloxone, I can actually obliterate the largest case in this aspect. So now we have a compound which is essentially active opioid agonist. Now the question comes is, what about the side effects associated with traditional opioids? So one of the side effects associated is tolerance. So now, at least in terms of tolerance, if I give morphine on day 1, and I give it for 9 consecutive days, and test on day 9, I'm going to have not the same level of effect I did on day 1. So I'm going to push the dose-response curve to the right. So morphine on day 1 is in red, which is shown here. If I give morphine on day 9, notice I produce the same compound is going to produce a rightward shift of the dose-response curve. And what you're seeing here is this hercanorin, which is in blue. If I give hercanorin on day 1 and I give it for 9 consecutive days and look on day 9, notice I have produced less tolerance relative to that of morphine in this case, which is nice and interesting. So it produces less tolerance relative to that of morphine. Now, a second case is if I give another way of testing sedation, all right, or motor coordination for opiates is actually that of the rotarod. And what ends up happening is this is a circular rod which goes on, and rats or mice are actually quite ambidextrous. And so what ends up happening is they actually stay on the spinning wheel quite well. But what happens is if you give a compound which affects motor coordination, they have a tendency to fall off the spinning rod. So if you look at the case here in terms of vehicle, rats or mice stay on the rod. In this case, if you give the mice morphine, what ends up happening is they fall off the rod, and of which slowly over time, they eventually come back to the 2-hour and it wears off the case. What you see here is that the hercanorin produces less motor coordination effects relative to morphine. So this is nice. So last case is how do you begin to address the abuse-related potential? And one of the ways to do that is what's referred to as conditioned place preference. And so what ends up happening is if you have animals which have, and give them 2 sides of a box, one being a light side or a covered spot, mice or rats, okay, by nature want to hide in dark places because that way it goes against, they don't want to be eaten by various predators. However, if one gives a rewarding substance, okay, in the light side of the box, they'll spend more time in the spots where they get the pleasurable stimulus. And so what this shows you is that if I give on one side of the box morphine, I produce a clear preference for one side of the box over the other as a measure of reward. Now, if I give hercanorin, I have no effect in this case because it's not centrally active. But if I give hercanorin, notice it produces less of a rewarding effect and produces less conditioned play preference relative to morphine. So what this means is it appears to have a less reduced potential relative to morphine in this particular assay. So what I'm showing you here is that we have a structural derivative which is different than morphine, which appears to have less, produces analgesic effects, which are blocked in an opioid-dependent manner, produces less sedation and less rewarding effects compared to morphine. So the question comes is, that's fine and well. Can we further push the envelope on these compounds? And so one of the things, at least that's been very important in the opiate field, is really the difference between codeine and morphine is the presence of this hydroxy group. So morphine is considerably more potent. And really, if we look at fentanyl and related compounds, are more potent than actually that of morphine-like compounds. And so the question came from a design standpoint, what happens if we begin to look at different substituents on the aromatic ring? Could we further increase the potency of these agents? And the quick answer is yes. And so notice now that if we make a judicious choice of a hydroxy methyl group, we can end up with compounds now which are even more potent than fentanyl, okay, in an in vivo sense in this particular fashion compared to that of morphine, okay? And furthermore, these compounds can be up to 250,000-fold selective for mu receptors compared to that of kappa receptors. Much like we saw with quercinorin, these compounds are centrally active, okay, and they can be blocked by opioid antagonists, as shown here in this case, in terms of antinociceptive. And furthermore, much to our delight, they also produce less tolerance relative to that of morphine in two particular types of tests compared in a different, slightly different motif in this aspect. So we're pretty excited about these compounds because we think we're really on the case of being able to have opioid-like agents that don't have some of the side effects seen with traditional opiates. But now I want to shift gears and talk about our other project we have going on in the lab, which is really to begin to combat the rise of fentanyl. So as I mentioned, really sort of the three waves of the opioid overdose death has really been the rise of deaths as a result of synthetic opioids. And namely the two ones that appear to be the culprits for that are actually going to be fentanyl, which is actually beginning to come in from China in this case. So what we've seen is sort of the difference now with the less of a dependent upon fentanyl or less of the dependence of heroin or actually that of methadone, and what we're now seeing is fentanyl. Now, the first thing I have to tell you about fentanyl, I mentioned this before, is that fentanyl itself is somewhere between 50 to 100 times more potent than morphine. This becomes problematic because the lethal dose of fentanyl is approximately 2 milligrams. And so what this slide is showing you here, here's a picture of a penny, and this is showing you a lethal dose of 2 milligrams of fentanyl. Now, it gets scarier from here because you can make structure-activity relationship changes, at least in terms of fentanyl, and you can actually go to a 3-methyl substituent and end up with a compound which is 10 to 15 times more potent than fentanyl. It's an analog called 3-methylfentanyl. Furthermore, if you make an additional substitution change, you can get O-fentanyl, which is approximately 30 times more potent than fentanyl. And the truly, really scary one is actually that of carfentanyl, in this case, which can be approximately 100 times more potent than fentanyl and almost 100,000 times more potent than morphine. And what we're beginning to see is fentanyl, 3-methylfentanyl, and fentanyl on the streets and are being cut and put into various types of heroin and is leading towards death associated with that. Now, these synthetic opioids are definitely highly toxic organic solids that are easily aerosolized and present a huge respiratory hazard. And this has been seen before because if you remember back to the Moscow hostage crisis, in the Treblinka theater in 2002, there was Chechnyan rebels took over 850 hostages in the theater. The Russian army pumped in a chemical agent trying to put people to sleep in the ventilation system to begin a rescue operation. Unfortunately, what they did is they aerosolized 3-methylfentanyl. And what this did is this actually killed 170 of people in addition to the terrorists in this act, and on top of that. So what they did is they succeeded in weaponizing fentanyl-related compounds in this case. And you can imagine a scenario where in the same type of chemical threat associated with these other sort of more potent fentanyl analogs producing a major chemical threat. Now, the only agent we really have able to combat the chemical threat associated with fentanyl is actually that of naloxone or Narcan. And there's 3 opioid antagonists on the market which reverse the effects of fentanyl and other synthetic opioids: naloxone, naltrexone, and Morphine. Now, unfortunately, there's emerging evidence that a single 4-milligram administration of Narcan is sometimes insufficient to surmount opioid toxicity associated with fentanyl. And in some cases, what this means is that the respiratory depression wasn't completely reversed and sometimes requires multiple administrations of naloxone to be able to do that, as well as other opioid effects reemerged after the naloxone dissipated. So indicating that we're not able to have compounds which are potent enough to surmount some of the opioid effects elicited by fentanyl-related compounds. And furthermore, the compounds are not long enough. So what this means is that we need longer acting agents related to naloxone or naltrexone. This work has been recently shown to be the case actually in rats and mice, showing here in this case. And what this is showing you is that naloxone is able to reverse okay? The effects of morphine respiratory depression, shown here on the top. This is a paper taken from the British Journal of Pharmacology, as well as notice that it's not as effective in eliminating respiratory depression elicited by fentanyl in a dose-dependent manner, okay? However, a different opioid compound, diprinorphine, which is an opioid antagonist, which is often teamed with actually that of atorphine, is able to overcome the respiratory depression seen from both morphine as well as actually that of fentanyl. So this suggests at least there's different mechanisms involved, at least in terms of the actions, and certainly between fentanyl related to that of morphine. But what this means is that if we have a different structural chemotype for opioid receptor antagonists, we might be able to better treat fentanyl-induced respiratory depression. And so one of the agents that we're doing that is to try to develop agents which are more potent than naloxone. One could argue in this particular case, if you look at the structure of diprinorphine, it's considerably greasier with a C-log-P of 2.57 compared to that of naloxone, which is 0.82. And so one of the things that we could do that is if we could make longer-acting compounds or more lipophilic agents, we might be able to better treat the respiratory depression associated with fentanyl-related super potent opioids. And so to this end, we've made a series of compounds and making analogs of naloxone and naltrexone to be able to using the general synthetic scheme shown here, where we can actually substitute things in the 14th position, as well as also changing the 6th position through a series of chemical transformations, which are shown here. So now we're going to go back and show you how we're going to test an antagonist. Okay, and so this is the same one we're going to be the case. So now, if we have sort of forskolin in green, it's going to stimulate adenylate cyclase to produce increased levels of cyclic AMP to produce light. If we give an opioid agonist, and such as fentanyl with an EC90 concentration, it's going to block the actions of adenylate cyclase through red, and we're going to produce this case. But now if I co-administer an antagonist in the presence of fentanyl, I can compete for the same binding point in a competitive manner, and therefore I can go back and see an increased level of light again to counteract in this particular case, to produce that. So to show you how this case, if I give fentanyl, as you might imagine, I will produce a potent EC50 value of 0.22, which is more potent than you see from that elicited by morphine. If I give naloxone, I can counteract the EC50, EC90 value of fentanyl with a value of about 60 nanomolar. If I go from naloxone to naltrexone, I can increase the activity about 10-fold to go down to 8.5. And if I go down to nalmorphine, I can further increase that. Now, one of the beauties here of medicinal chemistry, so one of the things we looked at was exploring the role of this 14 beta position substituent. And what we found is that if one makes a substitution here where they put a longer ring system with an aromatic ring, we can get a compound which is actually more potent than actually that of naloxone. But surprisingly, if one actually then looks at and changes the double bond, we can end up with compounds which are no longer antagonists, but actually now begin to switch and become agonists at the receptor. So I'll draw your attention here to one compound which has a particular naloxone compound. So one of the absolute tenets of opioid receptor pharmacology was that if you changed from a methyl substituent to a cyclopropylmethyl compound, you went to do that, you end up with an antagonist of receptors. What this is showing you is with a substituent here for the 14 beta, I can end up with a compound from naloxone, which is now even more potent than fentanyl is as an analgesic in this case, which is scary in and of itself from a drug design or from a synthetic substance standpoint. But furthermore, this holds because we know that we can make other changes, changing from a phenyl ring to a cyclohexyl ring. We notice we can get compounds which are exquisitely potent. One of the other things we notice, at least much to our delight, is that we can end up with compounds now, which by making changes here on the 14 beta position and the R1 position here, if we're going from the ketone to a corresponding double bond, end up with a compound now, which is 1 nanomolar as an antagonist compared to EC90 value relative to fentanyl. So these compounds are considerably more lipophilic relative to that, but we also have a compound which can end up being more potent relative to that of naltrexone, which is 0.3, which suggests, at least to us, that maybe that lipophilicity is not really a major determinant as to whether or not these compounds are going to function as good antagonists. Now, this work is ongoing in the lab, and we haven't really confirmed this yet, but I can tell you, looking here in terms of a traditional PA2 analysis, we can show you here in this case with a dose response curve here of fentanyl, shown here in black. Notice now that if we give naloxone in blue, we can get a rightward shift of the dose response curve, as you would imagine. We know that naltrexone at an equally potent and an equal concentration is going to produce a further rightward shift of the dose response curve relative to that of naloxone. And much are like compound 9 as well as compound 11. These two compounds produce a further enhanced rightward shift of the dose response curve, suggesting that we do have a lead towards the development of ultra-potent opioid antagonists, which potentially could be used to begin to combat some of the chemical threats associated by fentanyl and related synthetic opioids. So just to conclude with what I've tried to tell you today, hopefully I've convinced you that salvinorin and related neoclaridenes are an emerging class of CNS active compounds. They have the ability to treat pain with reduced side effects as well as drug abuse related events. Hopefully I've convinced you that quercetin and quercinol are two potent and selective, centrally acting non-nitrogenous probes for mu-opioid receptors with the potential to ease the opioid crisis. And hopefully I've convinced you that improved countermeasures are necessary and needed to better combat the threat posed by fentanyl and other synthetic opioids. And we have at least beginning to make some traction towards that end. Lastly, I couldn't have done this work all on my own. I need to thank my collaborators, Victoria University of Wellington, Bronwyn Cavell. The non-human primate work I discussed was done by Eduardo Butelman at Rockefeller University, and some other ones, at least from Kevin Freeman and related. And lastly, the really important people in my lab, which are actually those of my research group, which is my postdocs, Sophia Casket, Dan Lowe, Nazwa Boyohan, and my graduate students, Sydney Hedrick, Lindsay Kornberger, Kaylee Baker. And the work I showed you from quercetin and quercinol were done by my former graduate students, Rachel Crowley and Andrew Riley. And so lastly, I need to at least tell you, at least in terms of the stages of grief as applied to medicinal chemistry. And usually when one makes molecules and it doesn't work out exactly, you usually have some amount of shock and denial as to why it didn't work, which then just leads to some amount of anger. At some point you become the bargaining, which leads to just downright depression. And hopefully if you continue to make analogs, it eventually leads towards a resolution of the conflict and hopefully at some point over time, joy. Jeff, thanks again for the invitation to give a talk. And I'd be very happy to answer any questions the group has.
So it's my pleasure. Thank you so much. I'm going to moderate questions now.
Sure.
Dr. Johnson, go ahead.
Doctor, thank you very much for that presentation. My question to you might seem fairly out of the lab and more on the street type of a question, but from a marketing perspective, do you see some of these other antagonists other than naloxone becoming more useful and more utilized during this epidemic?
I think yes, and then we can certainly have a discussion as to whether or not that's really going to help the underlying problem of prescription opiate abuse, because it's certainly one could argue that by saving addicts with Narcan, you're just enabling them to continue to abuse, and that's certainly the case. But there are people who potentially want to be helped, and we certainly don't have, from the chemical threat perspective, these ultra potent compounds which are necessarily needed from the chemical threat. So I think it's a complex problem, and certainly I think marketing would be good, and certainly we need them to help, but certainly they're not going to be a catch-all or certainly a help for all types of situations.
Thank you.
You're very welcome.
Dr. Ogbas, you have a question? Go ahead.
Yes. Do you hear me?
I do.
Okay. Hi, good afternoon, Dr. Prisinzano. Thank you very much for bringing me back to 30 years ago that I was working on the kappa opioid receptor characterization in the rana esculenta and then switched to the human placenta. But that was 30 years ago. So now, as far as I know, these narcotics are mostly the CNS, central nervous system, especially for the the naloxone, the kappa opioid receptors and the mu. However, we were able to characterize them the human placenta. As far as I know, human placenta is a non-neural tissue. So that was 30 years ago. I'm just curious, so you are in the field, anything has changed? Why we can see the kappa opioid receptors in human placenta?
Well, we have
non-neural tissue.
Sure. No, we definitely know that opioid receptors themselves are both in CNS tissue and also neural tissue in this case. And so they definitely, at least in terms of pain, is we do see them, at least in terms of nociceptors, present in the periphery as well. And so one strategy towards the development of novel analgesics is to specifically target peripheral opioid receptors, in particular the kappa receptor.
And one follow-up question, if
Sure.
if the moderator kindly allow me. Then after finishing my doctoral studies, I came here for postdoctoral study, and we were working on the methadone replacement therapy, and then for the pregnant women who are in the methadone replacement therapy. And I was just collecting this blood placenta and then characterize the opioid receptors. But I have seen that the placenta was very much calcified for those patients. I'm just curious why.
That's a great question. I wasn't aware, at least in terms of the calcification of the placenta. We do see differences, at least in terms of some of the signaling properties in terms of opioids. So the different types of compounds definitely do produce different effects. And we even see that, as I showed a little bit, in terms of the data relative to sort of the respiratory depression elicited between fentanyl versus that of morphine. So whether it's an opioid-mediated mechanism for the calcification, or if it is just a specific component of fentanyl, I can't really tell you, but it certainly sounds interesting and something definitely to follow up on.
Thank you very much. Appreciate
You're very
it.
welcome.
Dr. Wolf, you have a question? Go ahead.
Yeah, thanks. I enjoyed this very much, and I'll actually throw out 3 questions, and you can choose to answer one or none, I suppose. But the first thing I thought about was a very interesting talk I saw at the experimental biology meetings years ago, or a few years ago, where Kirill Martinayanov, or whatever his name is, Mayanov, was demonstrating that opioid receptors actually couple to a range of G proteins, and that that arguably influence the responsiveness to things. The second question that I had related to your compounds, so your first one, the Kirk, whatever one it was, do animals self-administer that, and would it be possible that they would choose that over time if it didn't downregulate, or varying administration amounts? Or conversely, with your antagonists, are you worried about creating a hyperalgesic state by blocking the endogenous opioid regulation of pain sensation?
Sure. All great questions, and I'll try to answer all 3 for you. The first one is yes. I mean, I sort of simplified this a little bit because there is a whole premise dealing with what we refer to as biased signaling. What that means is that you certainly have compounds which are able to signal through the same molecule through the same receptor, producing different levels of second messenger signaling. In the classic sense, we sort of think about in terms of GPCRs, at least in the serotonin case, or whether it's a cyclic AMP mediated driven or if it's a phosphoinositol type of mechanism. And there are compounds which can select one pathway over another. And we don't necessarily know exactly the level at which those 2 things are going to signal. And there is some evidence which is suggested that if one could find compounds that don't signal through beta arrestin pathways relative towards G protein coupled signaling, you might come up with a better analgesic, because you produce less respiratory depression and less constipation and less tolerance associated with that. Now, this has been recently challenged through a series of papers, and I think the long and the short of it is we don't understand. But I think at least what it's showing you is at least the signaling pathways for molecules are more complex than we thought, and then you're going to see differences. So question number 1. Question number 2, in terms of self-administration, we haven't actually specifically looked at self-administration in this case. I would think on the basis of producing less reward, you might see less self-administration or at higher dose relative to the other opiates, but I don't know that for a fact, and we certainly need to test that. Now, in terms of the hyperalgesic state, yes, it is possible, at least with these super potent compounds, you could do that amongst the endogenous opioid system. But I guess we view these compounds as more of an application, not so much in sort of drug abuse, but more from the aspect in terms of a chemical threat, where we have a wide exposure to a chemical threat elicited by say, fentanyl-related compounds. Say somebody dumps a whole bunch of, of carfentanil into the water, and we need a quick, fast-acting antagonist to prolong life. These might be useful for that. Now, that doesn't preclude them from other types of applications, but I think we're looking more for just prolonging life quickly rather than sort of the hyperalgesic state, which certainly could be a case in terms of the antagonism of the endogenous opioid system. But an excellent question as well.
Question number 3, or did you already answer that? Dr. Schneider, your hand is up. Go ahead.
Tom, thank you. Very good presentation. I enjoy it. And I'm a immunologist, and your first time I saw after graduating 40 years ago from medical school, I saw a number of different formulas, etc., and diagrams. But let me ask you this question. So you presented data that methadone is not working for everyone, but nonetheless, in such countries like I know exactly in Sweden, it's distributed to combat drug abuse. So now, correct me if I'm wrong. Is methadone freely distributed in the United States?
Methadone is not freely distributed in the United States. One still has to go to a methadone clinic in order to get methadone.
All right. So
the big advance, at least in the U.S., was you can get buprenorphine, okay, outside of a methadone clinic, and actually can come from a pharmacy in this case, which makes it different.
Coming to a question. Of course, that's what you discovered, that you were able to synthesize quercinol or whatever it's called, right? So what the prospects of that drug to be distributed for people who are suffering addiction, right? So again, it's needed some formal, because I know in Stockholm they come, they get it, and they get out, and nobody asks who you are. Yeah. And at least they keep them from dying. That's what my question. What the prospect? What the future of your drug?
Well, the future is that we could that's certainly one tack that we can take. I mean, we have to certainly do a lot more studies to prove that it's effective in humans, and certainly so that the short take-home message is, if you're a rat and you're addicted to opiates, I have something to help you. And so I think through additional studies, we could potentially find a compound which could find some utility as a potential methadone-like agent. I mean, so certainly methadone. One of the advances, at least from a methadone replacement therapy, was the fact that it's much longer acting, 24 hours, versus sort of heroin or morphine, which is sort of on that 4 to 6 hour dose, which is why they have to come back each day. So you could supplant it, in that particular fashion, But I guess the short answer is that we're a long way away from that. We're certainly interested in, and that's certainly one of the things that we're interested in pursuing.
But that is primarily for professional use in clinics, right? So will not be. All right. So do you think that it is, what approaches from your perspective is more humane? What, let's say, Switzerland applied, all right? So what we strictly distributing methadone and in the United States? What's your take on it?
That's a great and loaded question, and I think I will take the fifth. I think you have to treat addicts humanely regardless, and there's obviously humane ways that you can do that. And so I think I don't think necessarily the United States is all right, and I don't think necessarily that Sweden and Switzerland are all wrong.
Tom, I bet Sweden will be the first taking quercinol.
Yeah, well, I might have to, probably ship stuff over there. So.
All right. Thank you. Great.
Very well. Nice visiting with you.
Okay, well, thank you, everybody. If there are no other questions, I will thank our presenter again for his talk and for joining us. And I will remind everybody that we do have a 1 o'clock journal club. Student Dr. Mitchell Fredeland, a second-year medical student from University of Colorado School of Medicine, will be presenting. So hopefully you guys can join us at 1 o'clock. Thank you, everybody. Have a good weekend.
Take care, everybody. Stay safe.